Okay, wonderful. Just wanted
Yeah.
to make sure the volume was okay. Well, thank you so much for the lovely introduction again. And I'm very, very sorry not to be there with you all in person as I had originally planned to be. And particularly sorry, given the reason that prevented me from being there is this escalating global conflict. And so I think the topic of this talk is particularly salient right now, that of international collaboration and how we can advance science, and particularly in this talk, our understanding of psychosis with this approach. So here I just want to briefly spend a moment to honor these really incredible, phenomenal folks, the graduate students, postdocs, and staff that actually make this talk possible, all the wonderful team members who are really great, a joy to work with, and also our international collaborators and the International Brain Behavior Consortium and our 22q11 working group. So that's my most important slide. So I'm going to start with telling you a little bit about what is Enigma. So some of you may have heard of the Enigma Enhancing Neuroimaging Genetics Through Meta-Analysis, many of these different working groups, and many of you may be involved. So in terms of understanding the genetics of brain structure, it's really important to pull together large samples for purposes of replicability and power. And so this Enigma consortium really started with my colleague Paul Thompson trying to combine data from essentially anyone in the world with an MRI scan and a DNA sample. So this really allowed the identification of the first genome-wide significant risk variants that influence neuroimaging traits. And now Enigma has expanded and diversified into a number of disease working groups around the world. And this kind of global collaboration and cooperation is really critical for advancing science, and I hope we can do more of it. So this is a map of all of the current sites involved around the world, involved in various Enigma projects and working groups, involves every continent except for Antarctica so far. And so there are really a number of advantages to this kind of team science approach in terms of statistical power, in terms of efficiency with distributed computing, and also collective intelligence when you have many, many minds who think in different ways trying to solve a common problem. So over 100,000 data sets, 2,000 plus scientists, 46 countries, and so forth. And so here we can see these working groups across different domains. So many different psychiatric disorders and neurologic disorders are represented, as one of the main questions across these groups is to see how diseases impact the brain. Several groups that are focused on methods development, as well as aging and development. And there are also many groups focused on genomics, which is what I'll be particularly telling you more about, as well as groups focused on infectious diseases. There's several new working groups focused on various environmental impacts, which just started. And there's probably more working groups that started, just popped up just since I started talking. So it's a very active collaborative enterprise. So these large international collaborations really have become particularly important for understanding genetic mechanisms of brain disorders. So as you're probably aware, neuropsychiatric disorders like schizophrenia are highly heritable conditions, but discovering the specific genes associated with disease risk has been a pretty challenging problem. Now, just recently, there's been major progress in terms of connecting genes to schizophrenia phenotype, finally, in these massive sample sizes, over 100,000 cases and controls. There are now, in fact, over 300 genome-wide significant risk variants in the latest genome-wide association study. Now, that being said, we still have a really poor understanding of mechanism. That is, our mechanistic understanding really continues to lag behind the pace of gene discovery. So how do we get from these genetic risk loci, these common variants of small effect, to this very complex and heterogeneous set of symptoms, delusions, hallucinations, negative symptoms, and so forth, that we call schizophrenia? We know our genes did not read the DSM, this lovely manual. So we have this big explanatory gap. And on the one hand, we know that these common variants can each contribute individually just a very modest degree of risk, and in combination possibly cause someone to reach the threshold for developing schizophrenia. But looking directly at that connection between the genes and this disease phenotype may not tell us what we want to know about mechanism. So one approach, one possible way forward is to examine these pathophysiological processes at the level of neural circuits, systems, cognition, and behavior that mediate between genotype and this distal heterogeneous disease phenotype. Now, in addition to the role of these common variants, there are also certain highly penetrant, large effect or rare genetic mutations, which confer substantially elevated risk for the illness. Compared to these common variants, you can see these are odds ratios. You can see the odds ratios down here. And particularly up here near the top is the 22q11.2 deletion syndrome, which you'll be hearing quite a bit more about in the next few minutes. And in particular, one of the values of such a model is that based on this known genetic etiology, we can develop animal models that have strong construct validity. The behavioral anomalies in the animal were really based on the same genetic defect. And they really can provide ideal systems in which to dissect circuitry mediating behaviors. And the other advantage for doing prospective longitudinal studies in humans is that these are mutations or conditions that can be identified very early in development. So I mentioned that the 22q11.2 microdeletion syndrome is a particular copy number variant, a particular chromosomal deletion that our lab has been really interested in for quite some time. It's also known as villocardiofascial or DeGeorge syndrome. And we initially got very interested in it because of its extremely high rate of schizophrenia. So what you can see here on the bottom, genome-wide, you can see that this deletion, the 22q deletion, really confers far and away the strongest association with schizophrenia of deletion copy number variants. Now, it's a rare disorder, but it's not as rare as you might think. It's about 1 in 4,000 live births. So it is, in fact, the most common contiguous gene deletion syndrome. There are some associated physical features like cardiac defects and immune deficiency. And again, the risk of schizophrenia is about 20 times that of the general population. There is also what I would call some diagnostic pluripotency in childhood. So they also have elevated rates of autism, ADHD, and anxiety disorder as well in childhood. So these are data from our multi-site consortium, the International Brain Behavior Consortium, which combined really allowed us a pretty decent sample size of patients with the same copy number variant, over 1,400 individuals. And so what we're looking at here is just the rates of psychotic spectrum diagnoses across the lifespan. And so you can see even in these really young children, 6 to 12, it's uncommon, but there are some with a psychotic spectrum condition. But across the lifespan, the diagnosis, the rate of schizophrenia, shown here in blue, it really tends to crystallize across the lifespan. And in fact, across these cohorts, 41% of the adults with this 22q deletion over age 25 had a schizophrenia spectrum diagnosis, which is quite remarkable. And so this is just to show you the demographics of the subjects that are included in the analyses that I'll be telling you a little bit more about across sites, Europe, North America, and South America in this analysis. And we had 537 with a molecularly confirmed 22q deletion, 331 age and sex matched control subjects. We also have a small sample with the opposite, with a gain at the 22q locus that I'll be talking a little bit more about at the end of the talk. So first, I'm going to give you a brief overview of the methods that we use. So to really think about how do these brain imaging indices map onto biology. So overall volume of brain structures is actually the product of two neuroanatomic features, the cortical surface area, or the total area covered by the cortex in a given region. Shown here, whereas cortical thickness reflects the average thickness of 6 cortical layers in a region. And notably, these indices are driven by different genetic mechanisms. They're both significantly heritable, but they're not actually genetically correlated with one another. And there's also evidence they're driven by at least partially different underlying neurobiological mechanisms with distinct developmental trajectories. The expansion of surface area is strongly impacted in early embryonic development via increased radial unit progenitor cell production. And this is from a recent, really landmark paper, actually, from the Enigma Consortium, a genome-wide association study of cortical structure, which found that the genetic variants associated with cortical surface area were indeed involved in regulating neural progenitor cell proliferation and cell cycle parameters during fetal development. On the other hand, cortical thickness, as I mentioned, really appears to be have a different underlying genes and appears to be shaped by later developmental processes, such as synaptic pruning and myelination. And so the methods that we use, I'm happy to talk more about them if you have questions, but essentially we have a very well-validated free surfer processing pipeline. This is the website here if you're interested, and we, in our analyses, typically covary for age, linear and nonlinear effects of age, as well as sex and site. And so now we'll get to some results. So here are what our cortical results look like. This is work led by Frank Sun here. And this is for 22q deletion compared to controls overall. So over here, we're looking at significant SMAPs for cortical thickness. And what's interesting is that we see an overall pattern of increased thickness in the cortex in the 22q deletion patients shown in blue, except for a couple of select regions, the anterior cingulate and the superior temporal gyrus, which is, of course, a brain structure involved in language. On the other hand, we see a very different pattern for cortical surface area where we see pervasive reductions in the 22q deletion group, really widespread across the brain. And in fact, the effect sizes of greatest magnitude are all in cortical surface area. And another thing I want to point out is that using a machine learning algorithm for classifying 22q cases versus controls, this actually worked extremely well. So the overall accuracy of this classification peaks at 94% when you put in 125 cortical features. And just for context, when you compare that to bipolar disorder case control classification, idiopathic bipolar disorder, that really maxes out at about 65%. So in other words, this is a much more consistent and homogeneous neuroanatomic signature that we're seeing. And in fact, if we just take our top two cortical features, that is the surface area for the cuneus and the lingual gyrus, we still get very good overall classification accuracy of 86%. So I think that's important to keep in mind. This disorder has a very consistent neuroanatomic signature. Now, how about the effects of psychosis on the brain? So for this analysis, we compared very well-matched groups of 22q deletion cases with and without a psychotic disorder diagnosis. And we found that there was significant cortical thinning in our 22q cases with a psychotic disorder diagnosis compared to those without. And you can see this is particularly in these medial frontal and frontotemporal brain regions, and more broadly in heteromodal association cortical regions. And so, and naturally, we wanted to look at the overlap with idiopathic schizophrenia. And so here we were able to work with Theo Van Erp and Jessica Turner, the working group leaders for the Enigma schizophrenia, who shared their data. And so given their really impressive sample size of over 9,000 people, you can see that almost the entire cortex is affected, but visual cortex is relatively preserved, whereas you see these larger effects in frontotemporal brain regions. And when you quantitatively compare, we do a cross-diagnosis correlation, we see that there is significant convergence with the cortical brain regions affected in idiopathic schizophrenia and 22q associated psychosis. So in other words, the brain regions most affected in 22q associated psychosis are really overlapping and converge with those seen in idiopathic schizophrenia. But interestingly, not so much for major depression. We really did not see a relationship between the brain regions most affected in major depression. So this relationship for the cortex is specific to schizophrenia. And so here, this is just looking across to give you a broader context of across disorders. And this is one thing that's really nice about Enigma is we can do these cross-disorder comparisons. And so you can see that 22q deletion really looks this is again 22q overall. This group looks really quite different from these other disorders in terms of the effect sizes and in terms of relative increased thickness of the brain, whereas most of these other disorders are associated with cortical thinning. Now, we also looked at subcortical structures, and subcortically, we saw some very quite large effects. These are Cohen's d effect sizes here, particularly for the hippocampus. And this was work led by Chris Ching, who's a former graduate student, now postdoc at USC, working with Paul Thompson. And one thing that was interesting is that these overall volumetric effects really sort of mask some of the more specific effects that we could see when we look at subregional differences in terms of subcortical shape. And so what we see here, and this is a method that Chris and others developed to look at subcortical shape, or we're looking at localized anatomy. And so what you can see here is that across subcortical structures, we see both local increases and decreases. But particularly the decreases that we saw were predominantly affecting subregions with projections to frontal, cingulate, and association cortices. Now, when we do that same comparison we did for cortex, where we look at the 22q deletion patients with and without a psychotic disorder, we see a very strong difference in terms of particularly hippocampal volume, but also with the ventral thalamus, where those differences were much more prominent, a much greater reduction in these structures in those with a psychotic disorder diagnosis compared to those without psychosis. And so that really, again, is consistent with what we see in idiopathic schizophrenia. And so again, we can do this really cool cross-disorder analysis where we look at convergence of how do these subcortical alterations in 22q-associated psychosis compare to other disorders. And interestingly enough, unlike what we see in the cortex, the general pattern of subcortical effects in 22q-associated psychosis actually resembles what we see for several other neuropsychiatric disorders. So these are the Spearman's rank correlations of effect sizes of subcortical structures most affected. Again, comparing 22q cases with and without a psychotic disorder. And we see there is a significant convergence with both schizophrenia, idiopathic major depression, bipolar disorder, as well as obsessive compulsive disorder. But interestingly, it did not overlap with the patterns that we see for ADHD and autism, which is interesting as these are childhood onset disorders. And again, we see across disorders the largest effects on the amygdala and the hippocampus. So what is going on at the genomic level? Well, we already know that this is a genomic hotspot region. It's very prone to mistakes and recombination because the region is flanked by these low copy repeats where you have the same genetic sequence repeated over and over. And this typical A to D large deletion, it includes about 46 protein coding genes. And what's interesting about the genes in the region, several of them are highly expressed in the brain and involved in early neuronal migration. And several that are involved in orchestrating self-fate decision in the cortex. What's become particularly interesting recently is the role of microRNAs, that is, these small non-coding regulatory RNA molecules that contribute to basic mechanisms underlying brain development and plasticity, and are really thought of as sort of master regulators of gene expression. And so before we even begin to think about specific genes, we really wanted to ask a basic question, which is the size of the deletion related to the severity of the neurobehavioral phenotype? And so again, as I mentioned, the most common is this large, approximately 3 megabase deletion. The smaller deletions, there's about a 1.5 megabase deletion, this A to B deletion, which is only in about 8 to 10% of cases. So as you can imagine, it's been tough to do an adequately powered analysis of this, but we're hopefully getting there. And so in the context of our large International Brain Behavior Consortium, what we found was that actually those with the smaller 1.5 megabase deletion, they have less genomic material missing. In other words, they have a significantly higher IQ relative to those with a medium or a large size deletion. So full-scale verbal and performance IQ were decreased by 0.3 to 0.5 standard deviations in those with the large deletion compared to those with the smaller atypical deletion. And so, of course, we were interested in whether this relationship might be linked to brain structural differences. And indeed, we do see that there are robust differences in cortical surface area as a function of deletion size, whereas the small deletion is associated with a significantly greater cortical surface area. And this comparison is here, the small versus larger deletion. So particularly in these medial frontal brain regions, you can see that there's larger cortical surface area in those with the smaller deletion extent. And this pattern does extend also to subcortical structures. Interestingly, we did not see this at the overall volume level, but when we look at these the subcortical shape differences, we do see indeed that there are significant differences, essentially an exaggerated pattern of differences, subcortical differences in those with the larger deletion. So mirroring the cortical findings, we see greater surface area of the hippocampus, the thalamus, and putamen in those with the smaller deletion. So now to try to address this question regarding gene prioritization, and can we get any closer to understanding what are genes within the 22q locus that might be driving some of these neuroanatomic effects? So we turn to an approach called imaging transcriptomics. And this is work led by Jen Forsyth, a former postdoc in the lab who's now at University of Washington. And so this is an approach that's become possible in recent years with the release of the Allen Human Brain Atlas and other postmortem brain resources, which are really phenomenal. This is a publicly available data set that has measured the expression of over 20,000 genes in brain tissue from psychiatrically healthy individuals, 6 donors, at a high spatial resolution. And using this comprehensive map of gene expression from human brain, it's become possible to identify correlations between the patterning of normal variation in gene expression across the brain and variation in brain imaging phenotypes of interest. And so this is just some examples of some studies that have used this approach, finding that among healthy individuals, variation in gene expression across cortical regions really recapitulates the spatial and functional cortical topography of the brain. And in clinical populations, for example, in Alzheimer's disease, this approach has been used to hone in on molecular mechanisms that may underlie brain phenotype. So here we're seeing variations in patterns of neurodegeneration is correlated with expressions of the tau gene, MAPT. And in schizophrenia patients, spatial variation in white matter disconnectivity is significantly correlated with the expression of genes implicated in schizophrenia by a genome-wide association study. So it's a really interesting approach. And we applied it here with the goal of really trying to understand or prioritize genes within that 22q locus that might contribute to neuroanatomic alterations. And so here we took, we used the structural MRI images from, we just took the 386 22q deletion patients that we knew had an A to D, the large same deletion breakpoints, 315 agent sex match controls, processed them with the same processing pipeline. And here we looked at the severity or the deviance of each region of interest for the surface area or cortical thickness compared to controls. And so essentially mapped that across the brain and then correlated that with the map of gene expression across the brain. And so here, this was the Allen brain data of 10,000, a little over 10,000 protein coding brain expressed genes. And so we correlate the severity of the deviance of either surface area or cortical thickness across regions versus the regional expression of each of these genes. And so again, here we're focused really on those genes within the 22q locus. And so what we found regarding expression of the individual 22q genes, variation in the expression of DGCR8, the DGCR8 gene in particular, was significantly associated with variation in surface area across the cortex. So essentially, in other words, the regions where DGCR8 is typically most highly expressed are the regions that show the largest surface area deviance or difference from controls in 22q deletion. Now, DGCR8 had the most robust association, but we also found another gene, a significant association between the regional expression of this AIFM3 gene, also within the 22q locus, and the pattern of surface area reduction among patients. So we were pretty excited that this DGCR8 gene popped out, as this is a gene that we know plays a major regulatory role via microRNA biogenesis, and it is known to be intolerant to loss of function and to regulate diverse gene expression patterns in the brain. So It's a key component of this protein complex that creates microRNAs, which are, again, these short non-protein coding RNAs that are involved in post-transcriptional regulation of gene expression. So as we know, DGCR8 expression is reduced in 22q because one copy of that gene is missing. And so we wanted to know which microRNAs show significantly reduced expression, and what genes do these microRNAs normally target. Now, unfortunately, we didn't have access to brain tissue from patients with the 22q deletion. There is brain tissue from mouse models, including a mouse model specifically of DGCR8 hemizygosity. And so to answer this question, we used a database from Joe Gogos's lab to explore the gene targets of microRNAs that are downregulated in cortex due to this DGCR8 hemizygosity, missing one copy of this gene in the mouse model. And we found that there were 59 microRNAs that were downregulated in the mouse cortex due to this DGCR8 hemizygosity, which actually targeted over 6,800 genes. And this is our gene ontology analysis, which revealed that these genes are differentially involved in regulating cell cycle parameters and gene expression. And interestingly, this is looking at this, the spatial or the temporal expression profile over time. And this red line here is birth. So you can see that these DGCR8 really shows its peak expression in mid-fetal development. And this developmental expression profile really makes sense given that cell cycle parameters regulate the rate of neural progenitor cell proliferation versus differentiation during early brain development. And so in any case, we are seeing this peak expression in early development. And these findings are correlational. So I do want to point that out. This is a correlation across the brain we're looking at. Sorry, my dogs are barking. But this association between DGCR8 and the surface area alterations really complements a growing body of experimental literature suggesting that DGCR8 is likely to play a key role in driving brain-related phenotypes. And most of this work is from mouse models shown here. But this also includes a recent finding from a collaborative study we did with Sergio Pashka's group on human-induced pluripotent stem cells from 22q11 patients. And she found evidence that the DGCR8 gene in particular was causally involved in defects in spontaneous neural activity seen in these organoid models. And so as we think about mechanisms that affect corticogenesis, a longstanding framework is the radial unit hypothesis, which was put forth initially by Pashko-Rakesh, which suggests that during early brain development, the expansion and size of the cortex is determined by this balance between cell cycle re-entry or exit, which determines whether neural progenitor cells will continue to proliferate or progress, and apoptosis or programmed cell death. So considering this, it's really interesting that the other candidate gene that popped out in our analyses was AIFM3, which happens to be a pro-apoptotic protein. And it appears to stimulate cell death by activating an apoptotic cascade. So together, this DGCR8 and AIFM3 are interestingly involved in these two neurodevelopmental processes that are thought to fundamentally regulate brain size and structure. So it provides us an interesting hypothesis regarding the potential basis for the prominent surface area alterations we see in 22q deletion. Now, in terms of how these 22q genes fit into the larger framework of neuropsychiatric disorders, this is again work led by Jen Forsyth. She used a method here called WGCNA, or weighted gene co-expression network analysis, to cluster genes based on their co-expression patterns. And here she used a different database called BrainSpan. It's a postmortem gene expression database spanning fetal development through adulthood. And so the first thing she did here on the left is to look across multiple data sets of both common and rare variants, that's including both de novo and inherited variants associated with schizophrenia and autism. And she looked at how expression of these genes clusters in the BrainSpan postmortem brain database. And so the first take-home point is we see that the 22q genes, shown here in green on the bottom, really cluster within these same modules as these other autism and schizophrenia associated genes. And then when we look developmentally, we see that these gene modules have different expression profiles across development. So there's this autism-associated M1 module, which includes genes, and then also this M7, which includes genes associated with both with schizophrenia and autism. They really are differentially, they are enriched for genes involving processes like chromatin modification, histone modification, regulation of gene expression. So really fundamental early processes. But then in contrast, we look at this M15 module, which is uniquely associated with schizophrenia. We see a very different developmental expression profile. Again, this red line here is birth. And so this is the module where the genes really peak postnatally. And it's involved in, and these are enriched for, this module is enriched for genes involved in processes like synaptic transmission and learning and memory. And notably, DGCR8 shows up here again in this autism-associated M1 module. So in the last few minutes, I just want to tell you about this other really interesting piece of the puzzle. So this was an intriguing, really intriguing paper that came out about 7 years ago from the Mike Owens group, presenting the first evidence that in fact the reciprocal 22q duplication, that is a gain of genetic material in the same locus, may represent a putative protective mutation for schizophrenia. And so they found this both in a discovery and a replication sample. And when I first saw this, I thought, well, that's amazing. Is that really, is anyone going to replicate it? And it turns out it has been replicated in the Psychiatric Genomics Consortium and also in a Han Chinese population. So in other words, if you have a 22q duplication, you are significantly less likely to get schizophrenia than someone in the general population. And this was a paper that came out in 2009 describing the 22q duplication as a new chromosomal syndrome. Now, interestingly, unlike the deletion, which tends to be de novo, the duplication tends to be inherited. So most individuals diagnosed with it have inherited it from a parent. Now, interestingly, a parent with the duplication may have a completely normal or near normal phenotype. The child may have an identical, will have an identical duplication, but may have obvious features like developmental delay, autism spectrum disorder. So it's not protective against everything. It appears to be just schizophrenia. So there are elevated rates of both the 22q deletion and duplication have been found in non-syndromic simplex autism spectrum disorder. So I'll tell you a little bit about our initial findings in the where we were able to compare these reciprocal copy number variants, the deletion and duplication. This is what our study participants look like. Both groups had a molecularly confirmed diagnosis of 22q deletion or duplication. Now, importantly, we did not recruit for any psychiatric phenotype here. The groups are really well matched in terms of age, sex, and parental education. But we see substantially elevated rates of autism as well as ADHD in both the deletion and the duplication groups. In contrast, we only see a psychotic disorder diagnosis in the 22q deletion carriers, as we would expect. And so this was work led by Amy Lin, a neuroscience graduate student in the lab. Oh boy, I'm sorry, my dogs are very excited about something. So when we take a broad look at the behavioral phenotype of the deletion versus the duplication, these are our Z-scores for different neurobehavioral domains in the deletion versus the dupe. And so with the lower scores being more pathological, this red is, they're all Z-scored to the controls. And so interestingly, we see that the subjects with the deletion have significantly lower IQ, but in verbal memory and processing speed, the groups are identical. And also in domains of social cognition and autism spectrum disorder traits, these are quantitative traits like sensory insensitivity, social reciprocity. They did not differ from each other, but on these schizophrenia-associated dimensional traits, we still see a difference where those with deletion have significantly higher, more elevated positive and negative symptoms. Now, what does this look like at the level of the brain? Well, in the context of other copy number variants, that is reciprocal deletions and duplications, the same segment of DNA, in other disorders, we've seen these mirror image phenotypes, which is really interesting. This has been found on chromosome 1q21, deletions and duplications associated with either micro or macrocephaly, respectively. And then at the 16p11 locus, a deletion at this locus causes a highly penetrant form of obesity, and the reciprocal duplication is associated with the opposite phenotype, is associated with underweight and reduced head circumference. And so more recently, the Simons VIP Consortium conducted a neuroanatomic study of the 16p deletion and duplication, and they find, interestingly, opposing brain phenotypes which differentially affected cortical surface area more than cortical thickness, increases in the deletion and decreases in the duplication carriers. So what does this look like for 22q? We also found opposing effects on cortical surface area and cortical thickness. Again, work led by Amy. And interestingly, we see these circumscribed differences in cortical thickness. So the duplications had decreased thickness, where we see in the same regions relative increases in cortical thickness in the deletion patients, and very widespread differences in cortical surface area. Now, across the cortex, this pattern was really the same, with the deletion showing the smallest surface area and the duplications the largest. But then when we, down here, these are Z-scored brain regions where we saw the largest effects. And so here what we're seeing is the brain regions that show the largest effects: the superior frontal gyrus, the middle frontal, the precuneus, the frontal pole, the cingulate, medial orbitofrontal cortex, the bank of the superior temporal sulcus. Any of those of you who study social cognition will recognize these as brain structures that are really critically involved in social cognitive neural circuitry and language. So this is really interesting that this is where we're seeing these differential effects on brain of these reciprocal CNVs. So that was what I showed, was just a snapshot of structural differences. But how about the role of neurodevelopment? Well, we know from this is a really beautiful paper that came out in Nature a couple of years ago, that we know that there are really significant neuroanatomic changes that occur across the lifespan, differentially affecting different cortical structures. And there's also a lot of non-linearity in these trajectories. So having these normative trajectories, we can then begin to examine how clinical populations differ from typical development. And so this is work led by Maria Jobzikowski, who is also giving a talk later on at this conference, I believe also by Zoom. And so I highly recommend that talk. But what we're seeing here, this is our longitudinal data. And interestingly, we are seeing that this pattern of cortical thinning is really more protracted in the 22q deletion carriers compared to both the controls and the duplication. So in other words, while the cortex, really the pattern really levels out in the other two groups, the deletion patients shown here in purple really continue to show this extended pattern of cortical thinning. And on the other hand, and we see the same actually for cortical surface area, where it continues to drop. On the other hand, the duplication subjects really do not show this same pattern. And so down here, we're looking at where over the course of development, where this pattern is most extensive, where the rate of change is most substantial. And so again, here we wanted to divide up those with psychotic spectrum symptoms compared to those without, who did not develop psychotic spectrum symptoms. And what we're seeing is that in those who the psychotic spectrum symptoms showed two distinct periods where cortical thickness that trajectory was altered. So really here in this early developmental period, and then also in this adolescent period, this is between the ages of about 16 and 20, we're seeing that those who develop psychotic symptoms showed greater cortical thinning during this time period. We really don't see this same pattern across for cortical surface area. So these distinct developmental periods where the cortex is really showing an altered trajectory in those 22q deletion patients with who develop a psychotic spectrum condition. And what was really exciting about this is that it aligns with the Enigma data, where we're looking at primarily at adults with this snapshot. So it really pinpoints this period in development when these cortical thickness differences arise. So we were excited to see that. And so just to summarize, the 22q Enigma has really given us the opportunity to combine data sets of this same copy number variant, the same 22q deletion across cohorts around the world. And we see that it has a really highly consistent and replicable neuroanatomic signature. The patterns of cortical and subcortical alterations in 22q deletion with psychosis converge with those regions that most affected in idiopathic schizophrenia, which is important because it suggests that these highly penetrant variants can inform our understanding of broader psychiatric disease risk. And we also see that deletion extent, those with larger deletion extent have more extreme cognitive and neuroanatomic alterations. We highlight a couple of genes, GGCR8 and AIFM3, that are involved in neurodevelopmental processes that are thought to fundamentally modulate brain size, potential drivers of this cortical surface anomalies that we observe. And we see that these common and rare variants associated with autism and schizophrenia really converge on biological processes related to early brain development and synaptic transmission. And also a new evidence that the 22q locus is associated with mirrored anatomic phenotypes that have divergent developmental trajectories and clinical outcomes. So of course, we've got a lot of work to do to really better understand mechanisms still. And so the ongoing work in animal as well as in vitro cellular models, we hope can really guide translation of more neurobiologically informed markers for therapeutic targets. It's a huge goal of this work. And so lastly, I just want to thank our funding sources, and of course, all of the patients and families who participated. So thank you so much for listening and for being here. I'm going to stop sharing my screen, and if there's questions, I'd be happy to take them.